Hey everyone, my name is Greg Lickert. If you're in the online portion of this class and haven't actually met me, I'm the TA for the World of Neuroscience course, and this week I'm going to be giving a lecture over kind of the fundamentals of how some things in neurons work. So that's where the neurons but zoomed in comes from, just like a zoomed in perspective, because the last couple of weeks we've talked about stuff like how neurons connect or some other different things, but today we're kind of focused on what exactly an action potential means and kind of some basics over how they're generated. And this was supposed to be recorded for the lecture I did in class a little bit ago, like today, but the recording didn't really turn out good, so I'm going to try it on this, and hopefully this is a lot better. So yeah, to get into things, a little bit about me. I graduated from a zoo with an undergraduate degree in biology, with an emphasis in human biology and medicine, with minors in computer science, neuroscience, and computational neuroscience. So I've taken a lot of courses. The whole point of me taking and being a TA for this course is that I can be a resource for you guys and I can try to help you guys out. I've taken a lot of courses here in the next couple weeks. When you guys start to sign up for courses, I'll come to class with a list of different courses that I've taken at Mizzou or that I know about that I think are pretty good. And if you are interested in one of the lectures, particularly, I can give you a course that maybe emphasizes that topic, or we can talk more about, you know, some people like to do like research, or we've had people in this class who start talking to some of the professors who lectured to help with that. The whole point of being here is to be a resource for you guys. So I just wanted to emphasize me being a resource and just a little bit about myself and why I might have a little bit of merit to give you guys a lecture. This is my fourth semester in grad school, and I've been to different neuroscience conferences and stuff. So I like to think that I know a little bit and can hopefully teach you guys something. So yeah. And then a quick side note, also before we get started on this kind of main lecture, this stuff is really complicated. There's a lot to it. We could depicted here. We have some different neurons. I just want to kind of show how complicated the structures of all this can be, and each one of the different branches might act differently and have lots of different stuff associated with it. So every time we talk in this lecture, and for most lectures that are given during this course, the professors kind of fudge things around or explain things not exactly how they work, but close enough how they work, because the true nature of how they work is really, really complex, and we don't really have time to get into that, and it might require a lot of background information. So we try to present things in a more easy to understand method. And this is another kind of example. This is an example of a spike raster. What that means is that each one of these dots is a different spike, and each color is a different type of neuron. This example has five different types of neurons. One of them isn't shown, but this is some stuff that neuroscientists look at and try to decode exactly what is happening. And there's a lot to even something like this over different rhythmic patterns in it and everything. So I just kind of wanted to show that this stuff really is complicated, and it's okay if you don't understand everything at first, but also if you ever take a more advanced course and you think back to something that we learned about here and you're like, that doesn't really sound right to what I learned in World of Neuroscience, it might be because we kind of fudge some things for simplicity's sake and to make you hopefully better understand those core concepts. So yeah, getting into the actual lecture, I wanted to talk about the structure of a neuron. So a neuron very generally has dendrites, cell body, also known as the soma, has an axon, and at the very end of the axon, we have this axon terminal. And so, kind of the order of how a signal goes is that it starts at a dendrite way at the tip here, and we'll have some kind of stimulus. We'll talk a little bit about what types of stimuluses things can be, but a stimulus, and then that stimulus causes some voltage change right along here, and that voltage change can travel along to the cell body through the axon to the axon terminal, where it triggers something, and we'll talk about that at the very end of the lecture. So something I wanted to emphasize with you guys, because I think it's pretty important if you take any neuroscience class, is how does the signal actually get started? So when you think of an action potential, something you guys might not know, or maybe you do know, but an action potential actually only takes place in the axon and some in the soma, but axon, axon terminal, and right around here in the cell, there'd be something called the axon hillock. And what that is that's the area that can actually trigger an action potential and cause that, typical spike that we've been talking about in class. So that's exactly like the structure of everything is very important and everything is very localized in neuroscience. So I think it's good to have an understanding of exactly what happens and where it's happening. So if we move on to the next thing, this is a example of an action potential or um, AP or lots of other things call it different things, but action potential is the most common. So we have here membrane potential. Membrane potential is the voltage inside of the cell. Our first lecture talked about this a little bit, and the voltage inside the cell, membrane potential, membrane voltage, different things like that. And we don't have units on this graph. The units would be right around here where this is flat and at a state of rest, so there's nothing really going on with the neuron, be around negative 70 millivolts. And the very peak of up here, we're going to call that around positive 40 millivolts. So different neurons behave slightly different. Normally, this negative 70 at rest can be plus or minus 10 millivolts, depends on the neuron and exactly what's going on. Same thing with the height of this action potential. It can vary also, but the general shape of an action potential is always going to be around the same. And hopefully you remember from the first lecture of the year how fast is an action potential. An action potential can take place in around 1 millisecond. So from right here to right here, about 1 millisecond. So it can act pretty quickly. But do neurons, that means a neuron can fire at 1,000 hertz or 1,000 times in 1 second. But that doesn't really mean that those neurons are firing at that rate all the time. I just wanted that to be kind of clear that most neurons in the brain might fire around 1 or 2 hertz or 1 or 2 times a second. And there is, that's what I'm saying, a majority of them. There is also, a big minority, so something like 20% of the cells or so, that might fire even higher. Some cells during a baseline condition might be firing at 25 hertz or something like that. So it's not unusual to have that as a high firing rate, but it's very, very rare, if ever, that you would see a cell firing at something crazy like 1,000 hertz or 1,000 times a second, or even something like 100 hertz. You might see different cells having interactions that when you see different graphs, you might have heard of something that's happening at 80 hertz in the brain or something. And that's never a neuron itself really firing that fast. It's a group of neurons, but that's a much more complicated topic for later. But I just wanted to kind of break down the fundamentals of exactly what this action potential is. And another thing that I'd like to focus on is how exactly do action potentials happen? What is causing this voltage to go from here to up to this peak, and then what brings it back down, and what causes that to have that pattern? So that's something we're going to talk about here. And then I just wanted to show an example here, and we're going to come back to this image as we talk about this lecture, but we can see that when we give a stimulus to a cell can either be something like a current injection, and that'd be something that a researcher would give. They would stick a like a metal electrode into you, and they'd shoot electricity into a part of the neuron, and you would record what happens. So here they shoot, you can kind of see that's when the flash, that's it's shooting, and you can see that it didn't cause an action potential, just a little bump. But then when we have a more aggressive stimulus, a higher current injection, then we can actually cause an action potential to happen. So this is kind of showing the all or nothing aspect of an action potential, that it's either there or it isn't. And so in this case, it hasn't triggered it yet, so this would still be read kind of as like a 0 state. And this action potential here, that would be like a 1 state. So even though there's a small bump here, there's no action potential, so therefore we call that as like nothing happening. So yeah, so how exactly do these action potentials happen? What is causing, when I give something voltage, in this case, what is causing the cell to have an action potential? And the key is ion channels. So ion channels are located all throughout the brain. Here are some examples of some. So we have a ligand gated channel, and what that means is that if we go to this little picture, that we can see this little dot right here, and that's going to be a neurotransmitter. So we can call that something like, you might have heard of dopamine before, or serotonin, those are kind of two big examples of kind of common culture things you might have heard for neurotransmitters. And what that means is that this chemical, like dopamine, is released and then it binds to this little channel right here. And when something binds to that channel, it is then able to open. Some biochemistry stuff happens, and then this channel goes from a closed state to an open state and allows allows ions to flow. So the other examples would be mechanically gated, which we talked about in our second lecture of the year, where the hair cells in our ears, they move to vibrations they detect, and then the hair cell deflects, and that deflection causes the the ion channel to open and allows the flow of ions. And we'll talk a little bit here in a second about exactly what flow of ions means, but for now, we're talking about exactly why they flow. They also have some type always open. These are also can be known as leak channels, and they don't have any. These other three types are gated by some means, meaning that they open and close given certain conditions. These kind of always open don't have that stipulation, and they're just always open, and they cause everything to kind of leak out slowly. And then finally, we have these voltage gated channels, and that means that given a cell voltage, that means that the channel will be open. So there's a certain channel that we'll talk about here shortly called a sodium channel, and those like to be open around minus 45 millivolts and will also then close at like 0 millivolts. So, and will also be closed anything below 45 millivolts. So we'll talk about some of these examples more, but something to note is that all of these allow ions to flow from areas of high concentration to areas of low concentration. So your neuron, you can think of it as like a big circle, and it's a wall. The cytoplasm, like the membrane cytoplasm, is on the outside, and the inside of the cell, and it's basically like a wall. And that wall is blocking this ion flow. But you need these channels to kind of open that wall up and allow ions to flow. And so you can have your body pretty much it makes a concentration gradient. So it makes certain areas have high concentration and other areas have low concentration. So if we go to this image, this might kind of help clear things up a little bit. During the rest state, which is nothing's really happening, there's a high level of potassium on the inside and a high level of sodium on the outside. Kind of an easy way to think about this is the salty banana. So bananas have sodium, salt, potassium, whatever. But during an action potential, sodium enters the cell, and that's just because, like we said, these ion channels allow flow from areas of high concentration to low concentration. So sodium is normally high on the outside, so it opens up and sodium comes in, and then potassium will have kind of the opposite thing. There's high potassium on the inside, so when a potassium channel opens, it causes potassium to flow out of the cell. And that flow of sodium entering the cell and potassium leaving the cell is what gives this action potential this characteristic shape. So, and then we see to get back to a resting state, we have this kind of transporter that takes sodium out and potassium in. But why exactly do sodium ions make this? Like, how do sodium ions and potassium ions factor into this looking like this? So, when you measure voltage, I'm saying this is negative 70 millivolts, and what that's in reference to is it's when you take a voltage of something, you need a ground. You need something you're comparing the voltage at the ground to the voltage at that site. So in neuroscience, you take the ground to be outside of the cell. So compared to outside the cell, the inside of the cell is negatively charged at minus 70 millivolts. And that is because there's lots and lots of positively charged ions outside of the cell. Now, even though I am saying that potassium, which is positively charged, is inside of the cell, there's vastly greater, more concentration of sodium and other positively charged things outside of the cell that make it so that the membrane potential of a neuron at rest is negative 70 millivolts. But then what happens is at the start of an action potential here, you're going to open a sodium channel. And when you open a sodium channel, what that does is it causes sodium to flow from outside to inside. And I'm saying earlier that the outside of the cell is much more positively charged than the inside. So when you let that positive charge in, it causes the potential of the cell to rise and go upwards, and that's called depolarization, making it less polar, meaning the difference between the outside and the inside is less. So it goes up, and that's because of that sodium. Then when the membrane potential gets to right here, that is the point when a sodium channel can open. And when the sodium channel opens, it takes positively charged sodium ions and it has them flow out of the cell because of that concentration gradient. There's way more potassium inside of the cell than there is outside. So potassium is going to go and it's going to leave the cell. And when that positive charge is leaving the cell, it then's going to bring that voltage back down. We have this little overshoot, which is something we want to talk about for now, and then it goes back to rest. And both those sodium and potassium channels are voltage gated, like I said. So the voltage for a sodium channel is going to open around negative 45 millivolts or so, and then it will shut again at around zero-ish, and then potassium likes to be open when the membrane potential is nice and depolarized. So the potassium will open here, and then as the membrane potential starts to fall, potassium will start to close, and then down here it's closed again, and then we kind of get back to a little baseline state. So there's also this little chart that kind of explains what I just said. So potassium opens here, that point to where there's kind of like a critical number of sodium channels that need to be open to cause an action potential. That's why when we go back to this doesn't cause a spike, but this does. It's because this voltage right here was not enough to recruit enough sodium channels to be open in order to cause an action potential. So that point when you get enough sodium channels to cause an action potential, here it's labeled as the threshold of excitation. There's lots of kind of different names for it, but basically once that voltage gets to there, there's pretty much no stopping that this action potential is going to happen. The cell voltage is going to go like this in this manner. And so that's a pretty important part when you look at a lot of things is what point does that cell start to have an actual action potential? And when that critical volume of sodium channels gets reached, it's kind of like a waterfall effect, and make it more positive, and it gets more positive, so then more open, and then it all gets shut off here. And it says here, around here somewhere at 2, potassium channels open, and potassium begins to leave the cell. And then at the very top, it also says that sodium channels close, and then kind of the opposite starts to happen, where potassium is now kind of the main driving force, and it brings us back down. So that's kind of like some basics over the action potential, and there is lots of different videos that you can find online that are going to probably explain this in a much better way than I am. But yeah, so that's kind of a very brief thing about an action potential. But something that's really important that is, that location is everything in neuroscience. Everything that's in the neuron is very precisely located, and it really does kind of change a lot of things. So for instance, action potentials don't occur everywhere in the neuron. They only occur along an axon, like I kind of said before. But why might that be? Well, the reason is because those axons, they have those sodium and potassium channels that are needed in order to cause an action potential. Something like a dendrite, which, just as a reminder, is kind of this part. It's the first step of where the signal gets sent into from an outside source like a synapse or current injection. These dendrites don't actually have the necessary channels in order to cause an action potential. So that's kind of why there won't be an action potential in the dendrites. It's because they just don't have the necessary tools in order for one to happen. So yeah. But in the dendrite, instead of an action potential, there's something known as a graded potential. So a graded potential is just like an injection of current here from like a synapse or something, and you can see that I guess is slowly leaving the dendrite until the dendrite returns back to -70. And that's because of those leak channels, so the voltage will enter, and then it kind of slowly leaks out due to those always open ion channels. Because anytime we talk about the flow of current, or I'm talking about voltage or anything like that, all of this is attributed to positive and negatively charged ions. So it's a kind of a bit of a different way of thinking about voltage and electricity and stuff that you might have thought of before, instead of how electricity in our, you know, in our house and stuff is different than electricity in our brain, because to the source of the positively charged ions. So yeah, gradient potential just looks like this. This is something that might happen in a dendrite, and then in the axon, we're gonna have something like this as an action potential. And so, get some different characteristics between an action potential and a graded potential. A action potential only occurs in the axons, and it's kind of an all nothing response. It's, either have an action potential or you don't. That's really what an axon cares about. And these signals, these action potentials can travel over very long distances. A lot of times in neurons, the axon can be really, really long and can go from one part of the brain to another part of the brain and communicate between brain regions. So those axons need to travel very long distances, and that's something that action potentials can do. Unlike graded potentials that, like I discussed earlier, will kind of slowly leak out and go back to baseline very quickly. Like it, it won't it won't travel a very far distance along in a dendrite. And these only occur in dendrites, and they also vary in magnitude. So these graded potentials a lot of times aren't going to be super high voltage like I showed here. This is in millivolt, this is like -56, but this is 40, so it's a very big difference. But if I wanted to increase the magnitude to the height that this graded potential reaches, I could um, increase the magnitude of the stimulus. So I could have either a stronger current injection or maybe a stronger synapse. Because in biology, I'm going to talk a little bit about some of that synapses here in a second, but a synapse is a connection between a neuron. So when you go and you look at it, there'll be an axon, and that axon will then form a synapse, and then it will connect to a dendrite. And those synapses can vary in strength. So you can have a really strong synapse, and that would cause a stronger graded potential compared to a weak synapse, which might cause a weaker graded potential. You can also have summation of these things in the dendrites. So you can see here a spatial summation, and what that means is that at the same time, we're going to have two of these synapses, synapse 1 and synapse 2, go, and you can see this response of synapse 1, this response of synapse 2. If we sum those two at the same time, it'll look like that, which is basically these two things stacked on top of each other. But you can also have something called temporal summation, which is when you have S1 and you have it happen, and then a little bit later you have S2, and you can see how it kind of goes like that, and then bumps up again because that second signal comes in. So you can have these things summed to form logic graded potentials. And yeah, they also, like I said earlier, can decay, which is what this graph is showing due to those leak channels. So yeah. And then something else I also wanted to briefly talk about was exactly how synapses work. So a synapse is a neuron is not directly connected to another neuron most of the time. I mean, almost all the time. And there is a little gap between them. So it's really hard to see if you're just like looking at a neuron under like a not crazy expensive microscope. But if you got a really, really expensive microscope, you would see that this is the axon terminal of one cell, and then down here is a dendrite of another cell. And there's a tiny, tiny gap in between. And so what happens is when that axon, at the very end of the axon, there's an action potential, and that action potential reaches here. Some business happens with some calcium channels, and it allows calcium to flow in. When this calcium flows in, if you are a biology major, you might have taken some biology classes. Calcium is a huge secondary messenger, which means calcium transmits messages in the cells. So calcium comes in and it tells the cells to take these things called vesicles. And a vesicle is just a sac of is just a sac in a neuron, and they are holding something. So here they're holding some neurotransmitters, something like dopamine, serotonin, glutamate. Those I think something like 100 neurotransmitters, but normally it's just one type. But it takes the neurotransmitter and it tells it to go to the very end of the axon terminal and then spit out the neurotransmitters. Then the neurotransmitters would be in this space right here, and then when they're in that space, they can flow, due to diffusion from high concentration, low concentration, lots from right here, low amount right here. They flow here, they touch this synaptic channel, and then that is a ligand gated channel like we talked about before, and will bind and will open. And in this space right here, there is a high concentration of sodium. And so then sodium will open up and flow into the cell and cause very large graded potential. And then that potential can flow all the way to the soma, to the axon hillock. And if it's a sufficient strength still at the axon hillock, then it will go ahead and it will cause a spike to occur. And so that is kind of the general flow of how a signal might go. Is it might go with, say, a dendrite right here, a signal bind, neurotransmitter binds, it allows sodium to go into the cell. Then, in that dendrite, you have this large graded potential, and this current will slowly travel down towards the soma, and it'll be losing strength due to it leaking out. But if it's a sufficiently strong signal, it will keep going into the soma, past into the axon hillock, where that increase in voltage can be sensed by a voltage gated channel, and then those voltage gated channels will then kind of take over the rest from there, and then they will cause a big old action potential to occur, and that really causes the signal to propagate and go down the axon, as action potentials flow to the axon terminal. And then when at the axon terminal, we get here, and if the action potential is still present, it will unleash some stuff, make these vesicles go, bind to this space, and then cause that same thing again. And this is happening all of your brain all the time. So that's really a lot to it. And so another kind of brief thing I wanted to discuss with y'all was this idea of the next chapter, or I guess unit of our course. Let's move over engineering aspects in neuroscience. So I just kind of wanted to tell you a little bit about an example of that. So I'm a computational neuroscientist. So what that means is I build mathematical models of the brain and then use them to study certain phenomenon. So if we look at this one, this is an example that our lab uses to try to teach neuroscience, and we have a whole course over all these that I'll mention to you guys a little bit later in the semester. But we can see that we have voltage, and when we change things like how strong the signal is, we should get some more stuff. I think I made it too strong there. Something like this. But we can also change the duration of it, and so we'll have more action potentials. And then we can change like delay, so those will fire later. And yeah. So exactly what this is showing is that you can take some guys in the 20th century who actually won the award in biophysics for this discovery, basically proved that you can take data from a biology lab, and collect data over how synapses work and how voltage gated channels work, all those different channels that we kind of talked about, and lots of others, and you can make them work. You can kind of copy the dynamics using math, and then we can have a computer do math, and we can make these kind of fake cells. So in my little fake cell here, we have an action potential occurring, and then we can see there's something called GNA and GK, and that's kind of like the flow of ions. So we can see at the start of an action potential, there's a very large flow of sodium ions, which we know is true. And then at the kind of the tail end of the action potential, there's a very strong flow of potassium ions. And then we can model other things such as currents, and even these things are called probability gates. It's not something we're going to talk about, but essentially you can model like exactly, like very, very zoomed in on those ion channels, exactly the characteristics that are happening. And you can take these models and we got one cell here, and we can take one cell and we can combine it and make two cells connect to each other. Then we can make a huge network of these cells and try to mimic actual brain tissue. So for me, I work on a model of the amygdala, which is something we'll talk about later in the course. The amygdala is kind of like the fear center of the brain, and we take this model and we try to do experiments that are otherwise very hard to do in biology, but due to kind of having total control of the situation, due to being a computer, we can kind of make everything go a little bit easier. So yeah, I just kind of wanted to introduce an idea of neural engineering, which you guys might not be familiar with, and we're going to discuss these topics and other kind of engineering topics in neuroscience um, starting next week. So yeah, hopefully you have a little bit better idea what's happening in the cell. There is lots of YouTube videos over this subject, so if you need help with anything, you can always reach out to me. But also, you can kind of do your own research and perhaps find a better video for how this is all going. So yeah, hope you have a good day.